Hello friends. So today we'll discuss about some very important enzymes which are used in molecular laboratory, that is DNase 1, RNase A and Proteinase K. You are surprised why we are discussing here today about these enzymes, because when you are working in molecular laboratory you should know the function of these enzymes, where to use and why to use and how much to use. So to answer all these questions we should study little bit about these enzymes. The first enzyme is DNase 1. DNase 1, as I have already explained you in the previous lecture, that DNase 1 it will chop DNase 1 what it do, it will chop your DNA in random by random cuts, and like it will cut here randomly and generate 5' phosphate and 3' OH, very small fragments. And where it is used, DNase 1 is used, as I have already explained, where you need when you go for the gene expression studies, where you need just pure RNA isolation. You are dealing with the in vitro transcriptions or gene expression studies. And then DNase 1, if you want to generate nicks in DNA, then you have to use your DNase 1. DNase 1 is basically it is extracted from non-animal host nowadays, because human cells also express DNase 1. So from cell lines or cells it is expressed, and otherwise DNase 1 is secreted by pancreas. So pancreas, and the gene for this DNase 1 is located on chromosome number 16 in humans. And DNase 1 is provided either 1 unit or 5 unit per microliter, like that. So in previous lecture I have explained you how much you have to use and for how much time, just to degrade your DNA. So we can recapitulate as, if you want to degrade your DNA so completely, that means if there is 1 microgram of DNA is present in your as a contaminant in your RNA, so you have to use 1 unit, and that unit will chop up your DNA into small, very small fragment, which will be further not be used as a template for the PCR. So this is your DNase 1, which I have explained you earlier in very detail, the protocol and all that. So you can go through that video also. The second enzyme is your RNase A, where we have to use RNase A. The second one is RNase A. It is also endo-ribonuclease enzyme. It will digest your RNA in between, and it will not digest, or it will not cut it randomly, but it cuts 3' end of pyrimidine, that is C or T. It will cut after C or T, so the RNA is degraded completely. So RNase A, where we have to use RNase A? So this endoribonuclease we can use when you are working with isolation of genomic DNA. So when you are working with genomic DNA isolation, there you have to treat your genomic DNA with RNase A. Secondly, it is used when you are dealing with plasmid or with the cloning and expression studies. So there the plasmids, when you isolate the plasmid, so the isolated plasmid should be treated with RNase A, just to remove your bacterial RNA from the bacterial or the host RNA from the eluted product. So how much to use? The concentration is, company people, they usually provide 20 milligram per ml stock, and you have to use 0.1 microgram per ml to 100 microgram per ml you can use in your reaction. So whenever you have to remove your RNA, that time you can use 0.1 microgram per ml to 100 microgram per ml of RNase A, and that is sufficient to remove your complete RNA from the solution. So this is all about RNase A. So remember that where to use and how much to use. So if you take just 1 microliter of your RNase A of having concentration of 20 milligram per ml, that is sufficient to remove your RNA. The third enzyme is your proteinase K. So and this RNase A, you will it is also available, or it is also extracted from bovine pancreas. So nowadays they are collecting or extracting from cells also, and as a recombinant also by cloning. And the third enzyme is your proteinase K. The third enzyme is your proteinase K. Proteinase K. Why it is known as proteinase K? The first is the proteinase K is extracted from a fungus, that is Triticum fungus, and from that fungus this is also endolytic enzyme, and this fungus from this fungus the proteinase K is extracted. And once you have extracted this proteinase K, why it is known as proteinase K? Because it can digest keratin protein, because it can digest keratin protein. That's why the name came from, the name is suggested as proteinase K, K for keratin. So it will, this proteinase has property to digest completely your keratin protein. And for the activity of proteinase K, it require calcium ions, and even it also require SDS, 1% SDS in the solution. So because this 1% presence of salts, the activity of proteinase K is enhanced, and it will chew all the protein from the elution at between 50 to 55 degree. If you have to keep your tubes or eluted products, and there you have to store, or you have to incubate for 10 to 15 minutes at 50 to 55 degree, just to remove all the protein when you are extracting DNA or your RNA. So there are nowadays many kits, even in proteinase K is used in both manual and kit based extractions of DNA. So there this proteinase K is included in the kit and provided by the kit manufacturers to remove all the protein, because all the proteins, they are the major contaminating stuff while you are dealing with your RNA or DNA. So this is all about all three your enzymes which are used in molecular laboratory. So in next lecture, what we'll learn very common reagents which are used in molecular laboratory, and that is TE buffer. I will explain how you have to prepare your TE buffer, and how much concentration, how much pH you have to set for that. So thank you for this. If you have any queries, you can write me, you can ask me, and I can help you. Thank you.